Hello, in this video we're doing topic 2.6, which is all about DNA structures in RNA. And in this video we're only going to focus on the multiple choice. We're going to see what kind of multiple choice that can be asked when it comes to the structures of DNA and RNA. So let's go ahead. So here we have the first question of DNA, which asks to distinguish. Now you can see the word distinguish here. The distinguish, what does it mean is tell me the difference between them. So that's what it means all the time. Now we have DNA and RNA, so we're trying to see what are the difference between them. Now we're gonna look at DNA and RNA structures here, a little bit of a scoop to remember it. So DNA, you know that is made of two strands, right? Because it's a double helix like that. While the RNA is only made of one strand, and it's basically a line like that. Now DNA, when it comes to nitrogen bases, it has A, T, C, and G, while RNA in this case has A, U, C, G. Now the sugar for the DNA is deoxyribose, right? While for RNA is ribose. So you can see that these are some of the differences they have structure-wise. So let's look at here and see if there's any differences on this chart. So for A, what we would say the DNA, the strands are symmetrical, while the RNA, they are antiparallel. Now, when you say that the strands are anti-parallel, basically you're saying that there's two strands and one is going this way and that way. So that already kills it because, first of all, the strands, DNA only has one strand, so it's supposed to be just one. So A is canceled. Then we look at B. Basically, DNA contains adenine, which is A, and RNA has cytosine. Well, okay, that can be true, but that's not distinguishing because they both have A and C, so it couldn't be B. Then C, you can say pentoses linked to phosphates and pentoses links to base. That's not true. Both of them have a phosphate linked to a sugar, and then from a sugar and linked to a base. So C wouldn't be the answer too. So the only one you have here, DNA is double-stranded and RNA is a single-stranded. So yes, the answer would have been D for this question. So moving now, because we're looking at differences, now look, this question asks the opposite. It asks for what is the similarity between DNA and RNA. So if we want to go back and look what are the differences, the similarities, you can see that both of them, if I highlight here, is that both have what? They have A, C, and G, right here, A and C and G. They both have the phosphate, right? And they do have a nitrogen base, but except for the U and T is different. But besides that, they do have a backbone that has a phosphate. So let's see what they are, the things that they say here. The first thing, it says both are polymers of nucleotide. That is true, right? Because what I mean by polymers, that means they have a nucleotide attached to a nucleotide attached to a nucleotide, and it just keeps doing. And it is, both of them is a chain of nucleotides. B says both are composed of anti-parallel strands. DNA, yes, it's composed of one strand going from a 5 to a 3, and this is going from a 3 to a 5. But RNA is just one strand. So B could never be it, the answer. C, both contain adenine, cytosine, and thymine. See, here is where the mistake is, because RNA does not have thymine, it has uracil. So C is out of the way. Now, both contain ribose. That's not true because ribose is just for RNA, and the sugar for DNA would have been deoxyribose, so it's not D. So the right answer here, it is A. So these kind of information, you have to go back to 2.6, and you really have to memorize and know the informations from DNA and RNA structures. Moving on to the next question here. We have hydrolysis of a pure sample of organic molecule. So we don't know exactly which organic molecule they're talking about, but it could be anything from carbs, lipids, proteins. So, but hydrolysis here, the word hydrolysis means is that you are breaking something down. And you're breaking it down from, let's say, a very large piece here of molecules, and you're breaking down into smaller pieces. So they say they produce a pentose sugar, thymine, guanine, cytosine. Now, who has a pentose sugar, thymine, guanine, cytosine? What other substance could be expected to be present in the hydrolyzed sample? Now, when you see thymine, guanine, cytosine, what is that? That is the T, that is the G, and the C. And it has a pentose sugar, which deoxyribose is a pentose sugar because this is what it looks like, a pentose sugar. So this is obviously is going to be broken down of a sample. Of what? In this case, DNA. right? And how did I knew that it was DNA? Because it said thymine, which is T, and it cannot be RNA. So if it's not RNA, you can always cancel the first one because it said thymine. And it cannot be B because uracil is just for RNA. The only thing I have left here is that we're talking about what are the substances present in this organic compound, right? So the only thing left here is basically the phosphate, right? So that will be C, the answer. Now, why would not be ATP? Because ATP can be found in anywhere, in any bonds that is going around. But we're talking about something that is composed in that substance. That means what is composed in DNA. And if we really look at the structure of a DNA, DNA is made of a phosphate group attached to a pentose sugar, which is attached to a nitrogen base. So you can cut and you will get phosphate, you can cut and get nitrogen bases such as the T, G, C, or even A. And then right here you have the pentose sugar, which in this case would be deoxyribose. So the answer here is definitely C. Now moving to the next question. Let's talk about the discovery of DNA because that's part of 2.6. You have to know how Crick and Watson receive credit for figuring out about DNA. So they want to know exactly what is the discovery that these two guys did about DNA. Now, they are the ones who took an X-ray, and in the X-ray, look at something like this. Not exactly this picture, but they took this X-ray right here, and from that, they created the model of DNA, which we know today, which is like this. Right? So it's a double helix. And this model is what these two men created. So what's the credit they made up? It was for the model that they created right here. So the first one says DNA is a molecule that the genes are made of. No, at this point, they didn't go in that details about DNA being the genes. The amount, B, is the amount of adenine equals the amount of thymine. That's the Chargaff's rules, which is not about them. The phosphate pentose bonding along with the nucleotides backbone is covalent. Now, that's not true, that they were not looking into what kind of bonds it is. They were just looking at what is the formation of the model. So they figure out the shape of DNA is a double helix, and that's exactly what it is. This shape right here is that it came out to be a double helix. So the answer for this is D. Now, some students might fall on the trick of this one, and it's not true. They didn't figure out what exactly the bonds. They just figure out that that's what we look like. Now the details, that's later on. Okay? Now moving on to the next question. Nucleic acids are polymers of nucleotides, and that's true. Polymers means a large pieces of units, right? So we have this attached to the next one, and you can take every single circle here as if it was a nucleotide, and they're attached to each other. Now that's a polymer, because again, the word poly means many, and the word mer means units. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 units. So it's many units. That's what a polymer is. Now, all of these nucleotides together, what do they make? They make a nucleic acid. Now, that's not what the question is, but we kind of get a little scope of understanding what was this concept. Now, it says what parts of the nucleotides are joined together in both DNA and RNA to make these polymers. So it's something that it can be found in DNA and RNA. So let's look. The first one says large nitrogen bases with small nitrogen bases. Some of you guys will say, well, I don't know, maybe it is true because the purines, they look like two rings while the pyrimidines are the small ones. And when I mean by purines, I mean the A and G, and the pyrimidines, it's T and C. So you might be a little bit doubt about this. Okay, let's go to the next one. It says nitrogen bases with hexose sugars. We don't use hexose. Hexose sugars is like this, right? And that is not what it is. Both RNA and DNA doesn't look like that. They look like houses. The only difference is RNA here has an O in here, and while the other one, DNA, doesn't. So DNA has no O in here, RNA. All right, and they do look like a house. So it's definitely not hexose. So B is out of the way for sure. Nitrogen bases with phosphates. If you remember how to draw a nucleotide, you will know that the phosphate is attached to the pentose or to the sugar. It is not attached to the nitrogen base. Here's the phosphate, here's the sugar. So how can you just say the nitrogen base is with the phosphates? That's not true. So it cannot be this one. And then it says the pentose sugars with a phosphate. And that is true, right? You have this phosphate right here connected to the pentose sugar. So D would have been the right answer. But we still are completely figure out about here. Large nitrogen bases with small nitrogen bases. When it comes to ATC and AGCTC, it's not really about their large and small. It's just about their structures. But it's not that one is way bigger than the other. So get rid of that idea, okay? So the right answer here is D. Next question here. It says, what does eukaryotic DNA have that is missing from prokaryotic DNA? Now, it's not uracil because uracil is all about RNA, right? So that can be killed. Now, what is it about? We will say the promoters in DNA. No, you do have promoters every time you're doing transcription on both sides. So promoters can be found in any kind of DNA, so it's definitely not it. Coding sequence, this one, every single DNA can code, right? Everybody has it. You have code sequences in prokaryotics, you have coding sequences in DNA. Now, intron here, it's a type of genes that do not express anything, right? They don't express. And this can only be found, that basically introns can only be found in the eukaryotic. You don't find that in the prokaryotics at all. So the answer is C. Now this is more like an HL questions, but I do like to put it here because sometimes, maybe makes the mistake of putting these questions. So know that introns can only be found in prokaryotics. And what are introns? Introns, they don't express, are genes that don't express. So that's pretty much very simple. 2.6 is very simple. Just really know the structures of DNA and RNA and be able to look at their similarities and their differences. And you know the DNA is all about the double helix. If you're good with that, then you have a very strong basis when it comes to 2.6. And that's pretty much for this video.